Hey everyone, and welcome. So today we're going to dive deep into some really important stuff, special use additives and procedures in phlebotomy. And listen, if you're gearing up for your NHA or CPT exams, you've definitely come to the right place. We're about to cover the key details you absolutely need to know to not only pass that test, but to really shine in your career. One tube, one draw. So what actually makes it the right one? Well, it's all about the details, right? In this explainer, we're going to peel back the curtain and look at the science of picking the perfect tube. We'll see how it ensures every single sample you collect is accurate, safe, and totally viable for testing. Honestly, this is where precision really starts. Okay, so here's what we've got on deck. First, we'll talk about why choosing the right tube is just so incredibly important. Then we'll get into the good stuff, decoding what's actually inside those tubes. After that, it's all about the unbreakable order of draw. From there, we'll talk about handling those specimens like a total pro, and we'll wrap it all up by nailing down those key concepts that you're definitely going to see on your exam. All right, section 1. Why do these special tubes even matter? Let's get right into it. You know, the tube you pick, it's so much more than just a little plastic container. It is the very first, and you could argue the most critical step you take to make sure your patient is safe and their diagnosis is accurate. And this is exactly why it's such a big deal. You know, making a mistake here isn't just a small oops. It can lead to some serious problems, like hemolysis. That's where the red blood cells literally get destroyed. Or it can cause these tiny little microclots that make the sample completely untestable. So getting this right from the get-go, that means you're protecting your patient from having to go through a second painful draw, and you're making sure the lab has exactly what it needs. Okay, let's move on. Section 2, understanding your tube additives. So what exactly is the secret sauce inside these tubes? Let's pop the top, metaphorically speaking, and decode the additives. We're talking about the specific chemicals and coatings that make every single tube unique and perfect for its job. First up, let's talk about the yellow top tube with SPS. As soon as you see this one, I want your brain to just scream blood cultures. The stuff inside SPS is actually pretty genius. Not only does it stop the blood from clotting, but it also helps any little microbes in there grow by kind of neutralizing the body's own defense system. Now, for your exam, and more importantly, for your patient, the one thing you have to remember is blood cultures are always first, no exceptions. This is all about keeping that sample sterile. Now, to make things a little tricky, you might see another yellow top tube, but this one's got ACD in it, and it does a completely different job. Its whole purpose is to keep the cells happy and alive. Think of it this way. The citrate stops the clotting, and the dextrose, well, that's basically lunch for the cells. You'll see this when used for really specialized stuff like HLA typing for organ transplants or DNA testing. And here's a huge exam tip for you. ACD is all about preserving cells. It is not for your everyday coagulation studies. Okay, moving on to the gray top. The magic ingredient here is sodium fluoride. Now, that sounds fancy, but it's an antiglycolytic agent. What does that mean? It just means it puts a stop to glycolysis. Basically, it tells the cells to stop munching on all the sugar or glucose in the sample. If you don't have this, the glucose level can actually drop pretty fast, which would give a totally wrong result. So for glucose or alcohol tests, the gray top is your best friend. And don't forget, it needs about 5 to 10 gentle inversions to get everything mixed up. All right, let's talk about the need for speed, because when you need a serum sample, you've got options. Your standard gold or tiger top tubes, they have silica particles inside that give the platelets a surface to cling to and start the clotting process. But that takes a while, about 30 minutes. Now, what if you're in a stat situation, an emergency? That's when you grab the orange top tube. It's packed with thrombin, a super powerful enzyme that gets that clot formed in just about 5 minutes. That time difference is a game changer when every second counts. So this table here, think of it as your ultimate cheat sheet. It lines everything up for you perfectly. The additive, the tube color, how to mix it, and the most important thing to remember for your exam. And hey, zoom in on that royal blue tube for a sec. This one's tricky. The color just tells you it's for trace elements. It doesn't tell you what's inside. You have to read the label to know which additive you're dealing with. Okay, deep breath. We've arrived at section 3, and this is a big one. This is one of the absolute golden rules of phlebotomy, the order of draw. And let me be super clear, this is not a suggestion. It's not a nice to have. It's an unbreakable sequence, and it exists for one simple, critical reason: to prevent the additive from one tube from contaminating the next one in line. And here it is, the sacred sequence. You need to burn this into your brain. Seriously, memorize it. It always starts with blood cultures to keep things sterile. Then comes the light blue for coagulation studies. You got to protect it from any clot activators. Next up are your serum tubes, then heparin, then EDTA. And bringing up the rear is the gray top. Sticking to this order every single time protects every sample you draw from cross-contamination, an error that could totally throw off a patient's results. Okay, let's talk about a classic scenario, one you are almost guaranteed to see on your exam. Imagine you're using a butterfly needle. See that long tubing? It's full of air. Now, if you go to draw a light blue top for a coagulation test and it's your only tube, what happens? That air gets sucked in first, the tube doesn't fill all the way, and bam, you've just ruined that critical 9-to-1 blood to additive ratio. The sample is no good. So what's the fix? It's simple. You just draw a discard tube first. It can be any tube, really, just to pull that air out of the line. All right, Section 4, handling specimens like a pro. You've done the hard part. You've got the blood, right? Well, not so fast. Getting the sample is only half the battle. What you do in those first few moments after the draw is just as important for getting a good result. Let's get one thing crystal clear about mixing. We are talking about gentle, smooth inversions, a full 180-degree turn of your wrist. Nice and easy. You never, ever shake a tube of blood. Shaking is like putting those cells in a blender. It's violence, it causes hemolysis. Remember, that's bursting the red blood cells, and it creates all this foam that can literally wreck the testing instruments. So what's the mantra? Invert, don't shake, and do it right away. Okay, so you've just drawn a gold or a Tizar top tube. As soon as that tube is full, an invisible timer starts. You have to give that tube a full 30 minutes to do its thing and clot completely. I know, it's tempting to rush it, but if you get impatient and throw it in the centrifuge early, the clot won't be solid. And you'll end up with these stringy little fibrin bits floating in your serum. And trust me, those little strands are the enemy of lab equipment. They clog things up and cause a huge mess. But what about those stat tests with the orange RST tube? Well, that's where things get fast. The wait time is way shorter, just 5 minutes. And you can thank that super efficient thrombin additive for that. But, and this is a big but, the rule doesn't change. You still have to wait for the clot to be completely formed before you spin it down. Whether you're waiting 30 minutes or 5, respecting that clot time is non-negotiable for getting a clean, usable serum sample. So before you send that sample on its way to the lab, I want you to become its final quality inspector. Just take a second and run through this little mental checklist. Give it a quick look. Is that citrate tube filled up to the line? Are there any tiny clots in that anticoagulant tube? Is the serum looking kind of pinkish or red? That's a huge red flag for hemolysis. A simple visual check like this takes 2 seconds and can save you and the patient a whole lot of trouble. And if you spot something wrong, don't even think about trying to fix it. The only right answer is to recollect. Okay, we're in the home stretch. Section 5, Mastering Exam Critical Concepts. Let's lock in everything we've talked about. I want you to think of these next points as pure gold for your certification exam. These are the high-yield, must-know facts that will not only show up on the test, but that you'll use every single day. Ready? Here are the non-negotiables. The light blue citrate tube needs that 9-to-1 ratio, gotta be at least 90% full. Blood cultures, what are they? Always first. EDTA tubes, keep them near the end to avoid messing with other tests. Gray tops, they're dead last. The royal blue tube, what's the rule? Read the label. And finally, invert gently, never ever shake. You get these 6 rules down and you are going to be in fantastic shape for your exam and for your first day on the job. So are you ready to protect every sample, every single time? Because by mastering these principles, understanding the additives, nailing the order of draw, and handling each specimen with care, you're doing so much more than just prepping for a test. You're stepping into your role as a true guardian of quality patient care. You are literally the first link in the chain of diagnosis. And in this job, every single detail matters.